One of my favorite ways to introduce my research to new students is to tell them I shoot lasers at stuff to see what happens. Once they're intrigued, then we talk about what is the technical language behind what we're doing, why are we doing that, what's the material, and for me, that's bacterial biofilms. My career award is about biofilm formation on medical device surfaces. So what that means is bacteria attach themselves to a surface. And as a mechanical engineer, I have access to a lot of tools and techniques to look at how well two things stick together. During my PhD, I studied the rules that govern how well things stick together. So no biology and pure mechanical engineering. Then during my postdoctoral fellowship, I studied structure-function relationships in our human cells and how those relationships differ between healthy cells and diseased cells. So now, as associate professor, what I'm studying is how well biofilms, so bacteria, how well they stick to a surface, and what are the rules that govern how well bacteria stick to a surface. So biofilm is individual bacteria that form this matrix, this surrounding goo, let's call it, and they live inside this goo, and that helps protect them from things like antibiotics, and it can cause destruction. 500,000 Americans have dental implants. And when bacteria attach themselves to the surface, they can form a colony. So they grow together, they create this goo, and it's really hard to get rid of either through antibiotics or through scraping. And so eventually, sometimes you get disease progression to where the implant actually has to be removed. What we're hoping to do is learn more about how these biofilms form on surfaces to create strategies to decrease antibiotic resistance and combat these medical device infections. People talk about their science being, the best product from their research. And for me, it's definitely my trainees. They are the best product coming out of my lab. And that comes with great frustration, but also such great proud moments too. So one of the most fulfilling moments for me was when we actually had something bad happen in the lab. We had some cross-contamination with the bacteria we were working with, and a student decided to take the sample to a scanning electron microscope, image it anyway, and colorized it and presented it to me. And even though, we're on this path of doing really rigorous science, we can learn from our mistakes, and we can move forward with that. To get to this point, that student had to, learn how to use this technique, had to acquire skills in doing the image processing and colorizing. And it was just such a great moment for me as a principal investigator to lead this team knowing, mistakes, they're honored and respected. They're inspected and respected as well. What makes UK a unique place to pursue this kind of research is that we have so many different colleges on campus from which to establish collaboration. So I collaborate with dentistry and pharmacy. I've got colleagues in agriculture. I've got colleagues in medicine. And this kind of research where we're approaching antibiotic resistance is going to take solving this problem from multiple perspectives.